So today I'm going to talk about Varroa mites, a little bit of the history, management, how we monitor them, and also how to treat. And then at the end, I'll touch on a little bit on what me and Matt do mainly here with the Bee Health Hub. But this introduction slide is two components that I feel are the most important for honeybees, which is food and forage, and then also varroa mite management. And on the left, you could see, a bunch of different pollens, and you could see here where they're starting to turn that into bee bread, where they're adding honey for lactic acid fermentation to store that pollen so it lasts longer. And then in this slide, you could see this mite just hiding right there. And there's two varroa mites here, and there's going to be a lot more pictures of varroa mites. But if you start seeing symptoms like this, we'll talk about that later. But that's when things start to become a problem. But know the pest. So basically, the varroa mite is sort of like an arachnid. It's got 8 legs, and it's in the genus Varroa, and there's 4 different species of this mite. And Varroa jacobsoni was named in 1904 in Java, Indonesia, and the original host was Apis cerana, the Asian honeybee, and South and Southeast Asia, and they evolved together, and it's not as harmful on Apis mellifera, that species, jacobsoni. So this was first reported in Apis mellifera in 1962 in Hong Kong, and then was later found in the U.S. and Florida in 1987. And you can just kind of see these localities here in the boxes in red. So is Varroa on Apis mellifera the same? Jacobsoni was found not to be able to reproduce on Apis mellifera, which is a blessing. So we don't have that pest as well with a different species. But Anderson and Truman in 2000 determined that Varroa is at least 2 different species of complex 14 haplotypes, which is a set of DNA variants on a single chromosome that tends to be inherited together. A little science mumbo jumbo for you, but only in Korea and Japan could these types survive and reproduce in Apis mellifera. And the species that causes all the damage was renamed to Varroa destructor, and that was also by Anderson and Truman. So, how did this shift happen? Not completely clear, but human movement is most likely, and I would attribute that in the United States and how it spread so quickly. It likely happened due to imports of Apis mellifera to Russia and the Far East. The mites jumped into new honeybee species and started parasitizing new hosts. And where are they now? Varroa destructor are spread across the globe by human and bee movement and people, maybe sneaking a queen and a couple bees on a plane or things like that. That's the way some of these can happen. Another way is on barges or ships. A swarm can move in. The people, moving these large shipping containers across the ocean, they don't know that the swarm's there. So a lot of prevention is done at the ports of import around these ships by setting up traps, and different agricultural units will be involved with that. A lot of times the USDA. So migratory beekeeping, shipments and queens and bulk bees is another possible way that these things can get spread around. And we also know that Australia has recently gotten Varroa, which some think it's been there for multiple years. and it just took a while to detect because they're not used to looking for it. So there's Australia, and this is basically the big news. Besides Tropical labs, but that's another mite for another day. So this is just a graph from Bee Informed, and this is a couple years outdated, but these are what beekeepers report killed their colonies or what they had issues with. And you could see that Varroa is pretty much number one on there. And then of course queen issues could be beekeepers smashing queens, could be bad mating quality. It could be a number of things. It's hard to put it all into just one picture and figure out exactly what it is. Starvation was a big one, which goes back to that forage that I talked about in the beginning. Some had no loss, which that was limited. And then of course weather, and then there's a whole list of other things that you know attribute to the losses. And when you mix all these things together in a pot, that's how you get the coin term CCD, colony collapse disorder, which is no single thing that causes the colonies to collapse. It's just a number of things that cause the colonies to deteriorate and eventually die or disappear. So why is Varroa so devastating? It's because it feeds on both the adults and the brood. And when it feeds on the adults, it's feeding on the fat bodies. And it also suppresses an immune response of the bees and reduces their survivorship by making their immune system more suppressed to these viruses. The viruses can really run amok in the bee's system. And once you have viruses, there isn't really anything you can do about it except try to control your varroa mites. And then this is an important one, mainly for queen producers, but everybody. It reduces your drone quality. So if you're trying to make splits and have the bees make their own queens, if you've got high varroa, there's a good possibility that you might be transferring viruses, or your mating quality won't be as good. But this all goes down to the main issue, which Varroa vectors viruses and spreads them, and it does it pretty well. So these are feeding site preferences for Varroa mites. And a lot of times people say, oh, I don't see any mites on the bees. Well, if you look at these markers here and where the mites hide, they're hiding underneath the tergites on the bottom. And then you could see the red dot on the left here. This is where they really like, is in between these segments, and you're not going to be able to see them when they're wedged up in there because they're feeding on the fat body. So if they're feeding, you're not going to see them. It's most likely like this dot, they're not feeding there, they're just waiting to hitchhike and get on another bee to feed, because maybe that bee is done, or they might be ready to reproduce and go into a cell. I don't often see them in this area here, but it's mainly the belly, or sometimes you'll see them in these back sclerites. Hopefully I'm getting that terminology right. It's been a while. But yeah, Varroa feeds on the fat bodies, and these are just some tests that they did, and it basically showed that using these dyes, that you could tell that the Varroa is feeding on the fat and not the hemolymph as thought in the past, which hemolymph is basically an insect's blood. So mites up close and personal. Idiosoma is this back, I call it like a carapace, like a crab. And then there's Gnathosoma, which is pretty much right around the mouth parts or sensory pedipalps. This would be similar to like a bee's mandibles or their proboscis and those parts. Very very similar, but this is again, eight-legged critter versus six. So I'm going to get into, if you did want to sample for viruses, you can, but as I said, there's not really anything you could do for viruses. You could just tell what the bees died from, and these samples can run a pretty hefty price, $115 to $150. Some may be even higher, but these are options if you wanted to get your bees tested. University of Maryland does it. Michigan State is setting up a program with Megan Milbrath. North Carolina State University just hired a new person, so they're getting set up to run more viral samples, which they've done a lot in the past. And then there's also the National Agriculture and Genotyping Center, and then the Apiary Inspectors of America's Diagnostic Lab. And you could just search those and find their websites and try to contact somebody on there. I didn't go through and try to figure out the pricing of everything. But to collect these samples, if you did want to do viral samples, it's very expensive as well. But you want to collect good and bad colonies to compare the difference. And these can be composite samples, which basically means you could take like 4 samples of good hives, 4 samples of bad hives, or you could do more, 8. But basically, you just want to fill a 50 milliliter falcon tube, which is, it's probably about 100 to 150 bees, depending on how many you could jam in there. A Ziploc bag would work as well. And you always have to collect the samples on dry ice. So this is where it gets expensive. Dry ice is expensive, and then the shipping of dry ice. You do need some experience to ship, and it's best to go to FedEx's website. That's what we use to ship these. But their website is very helpful, and you'll need dry ice stickers. But what the dry ice does is, it's at least negative 40 degrees, and it prevents the breakdown of RNA, which is used to identify the presence of these viruses and the levels. So now I'm going to get into some of these other viruses, and I've got some pictures of stuff, but I would say acute bee paralysis, these all kind of have similar symptoms, I would say. So, of course, you have this looking thing, which is K-wing, which is basically the frenulum, which is the hairs that connect the hind wing and the forewing. Um, they're disconnected for some reason. And this only has one side, so I wouldn't be too suspect. It's when you see both, that's when they would really call it K-wing, because it kind of looks like a K. But here you could see this is another symptom that people would call possibly a virus, but this could just be an old bee that the bees pecked the hair off, and you could see the wings are kind of pecked at. So it's just a picture of a shiny bee. It could just be an old bee. And then here again, you can see that just that one side where the frenulum is disconnected, from the forewing to the hind wing, because normally these are connected, which enables them to fly a little better. So there's different ways that viruses can be transmitted, and I think that the main picture of this graph, or whatever you want to call it, on the left, is if you look at the oral fecal transmission, it's a positive for everything. So, if you're seeing crap and things like that in the hive, if your bees are sick, you might think about wanting to do something about that, whether it be cull the frames and burn them if the bees are sick, or you could get them irradiated. But if you look at Varroa there, you can see that they transmit. There's 6 different viruses, and I know I'm missing some of the viruses here, but it's just important to know the different ways of transmission. And then you get your vertical transmission, which is, from the queen or mother, and from the father to the mother, and that corresponds with going to the worker that she lays, or the drone. So it's important, but again, just keeping your mite levels down enables you to not hopefully get all these viruses. So here's another virus. This is black queen cell virus. And this is mainly seen in the queen production industry. So for a normal hobbyist, or even in queen production, I've been doing this for almost 15 years now, and I've seen this one time where I've got that actual black larva. Sometimes guys will explain it as just the larva not developing at the correct time, so it's a delayed in molting, and sometimes they'll turn to mush, but sometimes that's European foul brood. So it's a tough one unless you see this actual black oily looking pupa in there, which this is a queen cell. And this is a picture from early on when I started, originally just right in 2011 here, working for Beam-Form Partnership. So sac brood virus is another virus which I saw this a lot more common in Pennsylvania. I saw it in Florida, but I do not see it that much in California, and I think it's because a lot of the beekeepers or queen producers that I work with, they all have really hygienic stock. So when these things are in there, they're plucking them out pretty easy because they kind of come out fairly easy if you grab them with the tweezers, but the bees can grab them with the mandibles and remove them. And I'll show you that in the next slide. But the transmission can be vertical, but a majority is horizontal transmission, which is bee to bee. The vertical transmission is from queen to daughter. So here you can just see right next to that sac brood virus is a Varroa mite just hanging out in there. And I thought that was a cool picture. But as this sac brood deteriorates, it'll turn this brown color. But usually, like I said, the bees will remove that before it gets to that stage. And I'll show you this second slide here. So here you could see you could remove it with the tweezers, and it is like a sac-like a pupa. It's just not developed, and I don't see it that often because again, it's working with these queen producers that have hygienic stock, and I don't see this virus very often. And if I do see something that looks like these, often what that is the early signs of chalk brood, which is another fungus disease, Ascosphaerus apis, but I'm not going to get into the diseases today. But this is one people are probably common with, is deformed wing virus, and transmission can be vertical, but mainly again horizontal, transfer from Varroa mite, or small hive beetle can actually transfer DWV when they're munching on larvae on the midline of the frame, and then the bees go to clean that out. They could pick up DWV. So yeah, and then you have your vertical transmission drones and mating and queen to daughter. And you could see some of these DWV bees circled in purple. And then another symptom that you've got mites is these chewed down brood or melted looking stuff. It's just the bees cannibalizing the brood. It's nothing different, shoot down brood and melt it are kind of the same thing. They're just cannibalizing. And here's more pictures of deformed wing virus. And, it varies in what it looks like. This one, a lot of beekeepers will call it stick wing. But you could see that some of the pictures, they don't look like sticks. But you could see, DWVB right by the queen could be feeding her that virus. And then I always like this picture because it looks like the bee is grabbing the mite off the back. One would hope, anyway. But here again is just some of that chewed down brood, and you could see that purple is actually a purple-eyed stage pupa that they're just cannibalizing it, sucking the juice out of it, and it could have died from virus, and they're spreading it. So it's very important to keep mites down so you don't see this stuff. And this is just more DWV because this is commonly the first symptom that you've got a pretty big problem. I mean, if you see one or two, that's a sign that you got to treat, and we'll get into that later. But if you see 30% of crawlers everywhere, that colony's 50-50 chance of living because it's probably got so much viruses in it that it's not going to make it through winter. So responsible beekeeping requires intervention. And bees are considered livestock, so not managing mites and controlling them, I feel personally that you should get fined if you're not controlling the Varroa and you're spreading them to your neighbor. But that's just my opinion. But parasitic mite syndrome is what happens if you don't treat for mites. And you could see in these arrows, all of them are mites in the cells. I mean, there's one there, there's one hiding right there, there's one on the side of the larva right there, there's another one hiding. See that little brown spot hiding under the capping? And yeah, there's just mites all through here. And if you zoomed in, there's actually one on the underside of the head of this larva that's kind of decaying. And then there's one right up there. So you're not going to see this when you're looking at the hives visually, unless like I take these pictures and then I can zoom in and find all these mites. But visually, I might find only those, the couple that are very visible. But it takes a while to be able to spot and identify that you've got a problem like that, except the melted brood. And this is just more on parasitic mite syndrome, and it's set by associated symptoms or signs. And basically the first thing you're going to see is you're going to see deformed wing, you're going to see mites on the workers, and the main identifier for PMS is when you see mites in the open brood. Sort of like over here, you got two mites in one open cell. To me, that's PMS right away. And you could see that it's melted, and then you got chewed down brood, and then some of this other stuff looks kind of funky. Another thing you'll notice is if you have high mites, is the bees will be pretty aggressive usually, and they're just mad. You'd be mad if you had somebody sucking the fat out of your body. I mean, maybe it would be a good thing depending on your weight, but for the bees, they're all the same size, and the mites sucking out the fat bodies is not good. She's not going to live as long. She can't fly as long. You'll see reduced population, and basically when you get to that form of PMS, the queen shuts down because they're unable to rear healthy brood, and that's really a problem. And the colonies will dwindle down really quick, and they'll pretty much disappear if you've got mite issues. And I'll show you some of the symptoms of what that looks like when a colony is collapsing from total mite infestation. But here's something kind of throw you off a little bit, but this is something that looks sort of like PMS, but this is called neglected drone brood. And this is when a colony might not have enough pollen stores, or they're just kind of on the verge of starvation. The first thing to go for the bees to take care of is these drones. And it looks melted. It looks like they're chewed down, and that's exactly what they're doing. They're cannibalizing this brood because they're not going to feed it, because it's an expense on the colony, and they need to concentrate on foragers to collect pollen and nectar. And here's just some more pictures of the neglected drone brood. And notice that it's mainly the drone brood. You rarely see this in worker, but you can see how it starts to look funky if the bees don't clean it up. It turns into this black scale looking thing, and some people might think that's AFB, but it's in drone, and it's not going to rope. And here's some more where it looks kind of nasty. And yeah, there's just a little bit more, but it's just something to show you that not everything is parasitic mite syndrome. There's other diseases, European foul brood, American foul brood, that could affect the colonies. But I'll show you some ways to monitor to know if mites is the problem, or you need to point the finger in another direction to try to figure out exactly what's going on in the colony. And that's one of my favorite things in beekeeping is I love looking at good colonies, but I like trying to figure out what's wrong with the sick ones, because that's what makes you a good beekeeper, is identifying and fixing the problem. So again, responsible beekeeping requires intervention, and I'm going to get into the treatments later, and I'm going to show you monitoring, which is intervention to try to figure out what your levels are to see if you need to treat. But these are just more stick wing bees, as a lot of the commercial guys would call them. But that bee doesn't look healthy compared to that one. Look at the size difference. It's a little shorter, and you could see that there's no hair on the thorax because the bees are picking on it because they know it shouldn't be there. So that's a symptom of DWV. A lot of people don't notice is they're often kind of hairless because the bees peck at them trying to get them to leave. So what can a beekeeper do about Varroa? And this is the integrated pest management pyramid of tactics for beekeeping. And you can use synthetic treatments. You can use other chemicals, which are organic or biorational pesticides like formic acid, hop beta acids, essential oils, menthol, and thymol. Not many people are using menthol anymore. And then oxalic acid, which we'll get into the treatments in depth at the end here. And then there's nematodes for small hive beetle and wax moth, which is BT. But if you have good colonies, you shouldn't have problems with wax moth, and that's again another story for another day. But I'm going to take any questions now. I know it's a little early on the break, but the next section will be a little bit more in depth, and I just wanted to give the history and some of the viruses and some of the things that can happen if you don't control Varroa. But next, I'll be talking about sort of the Varroa life cycle and the treatments, and how to monitor for them. But yeah, I'll take any questions.
I don't see any in the chat right now. Great content so far, Rob. I'm learning new things. Any verbal questions anyone would like to ask? Feel
Yeah,
free.
I mean, I know I'm a little early on the break, but it's just the way to. If I would have went into the next section, the break would have went
No, yeah.
too late.
There's questions are great. All questions are good. Anybody have a question?
Hey Rob, this is Matt. You could
Yeah.
mention There's a couple other ways I think to send viral samples. That was a good slide on where to send that stuff. I'm actually learning stuff here too. This is great. It had a couple different places. We usually send stuff to, Maryland, Beltsville, the lab there, but I don't think you mentioned, but there's also an RNA later product which people could use instead of
Oh, that
dry
is
ice.
that is true. So
Yeah, and then live bees too. I don't know if you wanted to talk about the other options that are maybe safer or cheaper, or maybe
yeah,
they're more expensive.
so RNA later is expensive, but we used to do that initially when I started the National Honeybee Survey. I think it was like 2009 we were using that. And basically what you do is you have a 50 milliliter Falcon tube with RNA later in it, and then you use the smaller tube, and you take your sample and you crush the bees up, and that preserves the RNA. Um, but the live bee sample is another possibility. But the thing is, when I talk to the people
Mm.
at the labs, the few people I could get a hold of, they said that they prefer it on dry ice. They don't want live bees. I
Oh,
Maryland might like the live bees, but you can get a small queen shipping cage, and basically you could make fondant or put fuller candy or whatever type of candy you want in there, and then you need a water source, which works really good with a sponge and a falcon tube or a tube, or you can just take a shop towel and fold it up and put it in a Ziploc bag and then cut a small square and fill that with water. And that's a good way um, for bees to get the water. But if you are shipping live bees, water is the most crucial thing. They can go without food for a couple days, but the water they really need if they get warm. So the cheapest way is a Ziploc bag with a shop towel folded up, and then cut a small square with a
mm.
razor and fill that with water. Um, but yeah, thanks for reminding me about that. I didn't get to talk to people at the labs in time to figure out how they wanted the samples, but the few that I did, they were doing dry ice and um,
Mm.
yeah, but
Yeah,
thank
we've
you.
we've done all those. Yeah, I know there's a project going on with the Sacramento area beekeepers where maybe they're going to be able to take viral samples. I don't know if Rick is on, but yeah, there's different options for sending those samples to a lab. Unfortunately, we're not doing viral analysis at the UC Davis lab yet. Um,
Yeah, and there are other labs that do viral, but not for the public. So Minnesota, Declan does stuff,
mm.
but it seems like he's mainly working on scientific projects, which I get it. But yeah, I mean, the
And
samples are expensive. What do they really tell you? When I started this job in 2011 out here, we took viral samples of every breeder that we
yeah,
did. And then, after a couple years, they're just like, we can't really do anything about viruses, like where are we killing more bees? Because we're already taking a sample for Varroa. At that time, we were sampling in a 4 gram bottle and not doing alcohol washes in the field. But
mm.
um, yeah, the queen producers and other commercial beekeepers, they just felt like if you don't control Varroa, you're gonna have viruses. So the main name of the game is keep the Varroa low. So
Yeah,
um,
it is. It's not cheap. I think I saw it was Maryland is charging $144 now for
yeah, I
vinyl.
saw like
Yeah.
150, and
Okay.
and I think there's one guy, he uses a machine, and it's not respected in the science community, so I didn't mention it. But he does samples a bit cheaper, and you just send them in a bag, and they don't have to be on ice.
Oh,
But if you guys wanted, I could get that information if you're interested in viruses. But to me, keep the mites low, and then you don't have any issues. But So Wendy and Kiana, do we have the 10-minute break now, or should we keep plugging on?
Sure, it's up to you. If you feel like 10 minutes, we could do 10 minutes.
Yeah, let's give people a break, and maybe they'll do like a 5-minute break and then come back. Maybe they'll have some questions. I was really hoping to have more questions. I know there wasn't a bunch there except for virus and history of Varroa, but you gotta start somewhere, and starting at where they came from, I think, is important.
Sounds
Right.
good. We'll take 5 and see everybody back here at, let's go for 9:50, shall we?
Yeah, and come up with some questions. Try to stump me here.
Oh,
Matt, that includes you. I'm fine with it.
challenge accepted.
All right. They're right. Insects. Are the spheroids on the insect from the top or the bottom? Are the spheroids on an insect on the bottom? Hey, Matt, you there? Or Wendy, I guess early. I like the laser.
I'm curious, Rob, to know if you remember the highest mite count you've ever found in a colony out of all the colonies you've been in.
I think the highest one was 167,
Gosh.
but
Wow.
I know other tech team members that have gotten higher. I think Catherine had something in the 200s.
Wow. Wow. It's amazing that bees with that size of a mite load just don't abscond, or maybe they're just too
Well, the crazy
parasitized to even think about it. They
Well,
just,
the thing was is there was a ton of brood, and if you looked at the colony, you wouldn't think that it had that many mites, but it
wow.
did. And this was a stack up, so it was what they do when they're done finishing breeding. They take the mites from the mating nukes and they put them in these things called stack ups to preserve the comb and keep brood on it, keep bees on it, less problems. But the mating nukes are often a source of high mites by, the fifth, sixth, seventh round of catching queens is when they start absconding because of mites or heat. So some beekeepers that raise queens longer, they actually treat the mating nukes. Um, but this is a cool picture here. I thought this is from Southern California. This is a holding yard before they go into almonds. And you would never keep that many colonies in one yard. But you can imagine uh, what a nightmare of robbing that could be with how many are there. I mean, there's just set after set after set.
Yeah, and how much it would cost just to feed them, because you can't just pop them down there without feed before they go to almonds, right?
Yeah, yeah, and they tried to keep them on the flat, but
Yeah.
you got to watch when you're filling them with syrup on a hill because you can't fill it up as much.
Mm. Mhm, mhm. Yeah. Cool. Well, we're 9:51. We'll wait till Keon comes back so he can press the record button.
Yeah, and was there any more questions that came in? I know
No,
that first section, it was just the virus stuff. The next two sections are my favorite. The life cycle of Varroa is not super interesting to me, but it's important to know as far as your treatments and things go. But I'll go through a couple of slides of that.
yeah, let's do it.
Yep,
Yeah.
recording is
I had a
already
quick
on.
question.
Yeah,
So I just wanted to know if anybody here knows the progress of genetically producing the more of these Varroa sensitive hygienic bees to
Is
where
it
it would cut down on maybe cutting down on the need to treat and loss of bees. Just
And
in,
there is later in the presentation, I will touch on some of that stuff. There are some genetic lines coming out of Baton Rouge, Pro line. They used to come out of Hawaii, but they it's pretty interesting. They started off, I think, in 2012, and what they did is they got different lines of bees and they put them through the pollination circuit that they run on the East Coast. And then they also did one in the Midwest. And then they selected the ones that had the lowest Varroa out of that. And that's how that pole line actually got started. So I just learned that from Garrett at Baton Rouge Lab. He works for Frank Rinkovich and does all the artificial inseminations for the pole line.
right. Yeah, because I noticed I requeened some of my colonies with some from a reputable a company here in California, and I noticed that my mite levels were down, and also I when I pulled the capped brood, when I pulled a frame of capped brood, I would notice that some of them were partially uncapped in the center. And that to me, maybe I'm wrong, but that to me indicated that there was some hygienic behavior going on within that
Yeah,
colony.
yeah, and that's a prime example of Varroa sensitive hygiene or VSH. And what they do is they're uncapping because they detect the Varroa, and then that disrupts the reproduction, so that mite won't reproduce. But it is important when you see that, it has to be a perfect circle in the center to know that they're recapping it, because if it's jagged, that means that it's probably a pest, or the pupa died of a virus or disease or something. But if it's a perfect circle, you can almost guarantee that they uncapped it and they're recapping. And Matt, maybe you can refresh my memory on when you do flip the capping over, there's a way to tell if they were recapping it or if it was just the bees pecking into it. I'm not sure if Matt's here.
Anyway, that answered my question. I appreciate it.
Yeah, and we just started this year doing the VSH assay, and we've done quite a bit, but the mite levels were so low, it was really hard to find non-reproductive mites. So with that VSH assay, you're actually uncapping 100 to 200 cells, and you're looking for just one foundress in there and not a little mite family, which would mean that they're reproductive. So the main thing with VSH is you're looking for the non-reproductive, which means that they open the cap, like you were saying, and disrupted that mating, and they weren't able to replicate or reproduce.
Great, thank you.
Yeah, for sure. Thanks for the question.
Okay. I think we're ready to continue if you are, Rob.
I am ready. And I know when we tested this out, I had the thing that would circle, but I do like this laser being able to point. So I'm excited for that. So what is integrated pest management approach? Basically a decision-making process based on understanding host and pest biology and host-pest interactions. These are basically based on thresholds by monitoring, which I'm going to get into in detail. And then also, using multiple different tactics by varying your treatment. So you're not just using one treatment to treat mites all year. It's best to alternate, so there's no chance of resistance. And if there is resistance, you're getting those mites with treatments they may not be resistant to, and ones that they are resistant to, you're using another treatment that they're not resistant to, if that makes sense. But you're trying to find the safest, most profitable way, and environmental friendly way, to keep bees. And one of them is, as you had mentioned, I'm not sure if folks were listening in, is like the Varroa sensitive hygiene, where the bees are controlling the mites on their own. And I'll get into that a little later. So let's learn about the Varroa life cycle. This is kind of important with your treatments, but it starts with an adult bee, and a Varroa is feeding on the hemolymph. And once that mite feels like she's fed enough, what she does is she goes into the cell right before the cell is capped. And you wonder, how does she go into that cell before it's capped? She can smell the pheromones that the larva puts off that lets the workers know that it needs to be capped. So what she does then is she goes into the cell and she goes into the bottom, as you can see right down here, and there would be royal jelly there, and the Varroa mite actually has the mouth part. It's a snorkel-like apparatus, and it almost will like lay on its back or its side, and it breathes through that snorkel-like apparatus to hide from the bees before it's capped. And then once the cell is capped, then the mite comes out to feed on the prepupa, and you could see the mite feeding here. And then the female will lay her first egg about 60 hours after the cell's capped, and then she lays eggs pretty much at 30-hour intervals after that, which could mainly male and female. 1 to 6 eggs develop to larva, protonymph, deutonymph. And I have some pictures of these different stages of the mites here in a few slides. But the developing mites feed on the bee damaged and leaving the bee exposed with open wounds, for different things to get in. At 7 to 8 days, you get an adult female. At 5 to 6 days, you get an adult male ready to mate, and then the mating begins within the cell. I've as I said, we were doing the VSH assay, and sometimes you'll find 2 foundress and a worker, and sometimes even more in the drone, 3, and I'm sure there's been 4. I haven't seen it personally. But when the adult female leaves the cell with the emerging bee, she's basically hungry and needs to feed on that bee to repeat the process. And the male and the immature stages, they basically still stay in the cell and die. And this goes back to that picture where I said the mites on the thorax, they're ready to transfer. That's what this number 10 is. The mites transfer by close contact. I've actually seen in a lab setting where the mites would piggyback on each other so they could get higher to try to reach another bee, which I thought was interesting. But a little more about the life cycle. The gravid female, which means pregnant, enters the cell right before it's capped. I don't know if that's but starts feeding on the larva and prepupa, and then, like I said, the first egg is laid at about 60, 70 hours after capping, and once the haploid male is produced first, that's the one that's going to mate most likely with the female. And the foundress keeps the wound open on the pupa so they could feed. And the foundress is the first mite that went in, or she's like the queen pretty much of the mite. And you can imagine each mite foundress, there could be hundreds in a colony. So there's like hundreds of queens inside, which is basically a foundress. And additional female eggs are laid about, like I said, 30 hours after. So she just lays a certain amount of eggs, and she's hoping to get at least one daughter that's going to be able to continue the cycle. And the eggs are laid on the cell walls, or I've seen the eggs also laid in the bottom of the cell without a microscope or at least 3.5x magnifying glasses. You wouldn't be able to see the eggs. They're very small. Even the male mites and the female mites are very hard to distinguish at the early stages. So it goes protonymph, deutonymph, and then adult. And here's kind of some images of what I'm talking about. So you see these tiny eggs all the way on the left, and then it kind of progresses going to the right. And you could see that they look very similar when they're small, between the male and the female. It's only once they get to the deutonymph where you can kind of distinguish them, because the male's legs are a little bit longer, and the ideosoma, I believe, or I call it the carapace, which is like the back plate, where it's more round on the male than it is on the female. But you very rarely ever see the male mites. You will see Varroa in alcohol washes where their exoskeleton is very light color. They're almost clear, and that's because it hasn't become sclerotinized, which is basically the chemical transition of the exoskeleton becoming hard, and that's what gives it that red. When it's like clear or very light, that exoskeleton is very soft, and they're kind of vulnerable at that point until they get sclerotinized. So here's kind of the mating behavior, and I don't really have any good pictures of the mites reproducing in the cells, which I should from the VSH stuff, but I'm sorry I don't have those to include. But mites do have a preferred defecation spot, and this I have pictures of, and it's usually on the top of the cell. And let's see here. I'm just going to show you. Here's a mite. This is PMS classic. They're kind of chewing down that brooder. It's melted. And then here you could see this was a larva that didn't have a developed head. And when they cannibalize it, it looks like this melted glob. But you could see that mite is in there still feeding, and that would be PMS, which I'll go over. And here's just more mites in the cell. And then there's the face chewed off of an older pupa. And this is often a symptom. If you see, 10% of that going on in your hive, you've got a mite issue. And as the gentleman had asked about the bees that can fight off the mites, this is what I was talking about with the cappings. You see how these are kind of jagged. This is what I was talking about, where they're actually going in there because there's mites and there's issues where if they were recapping this, it would be perfectly circular, where these are kind of chewed down and jagged. I mean, that one looks like they might be recapping it, but it's just kind of too jagged for me to make that prediction. So this is what I was talking about the defecation sites. You can see the poop right on the top of the slides, or frass, but this is a colony that most likely probably died from mites when you're seeing this frass on the top. And I do have one more picture, and you could see it here. That white is not mold or anything. That's actually the frass from the mite, and you could tell there's more than a couple cells that had mites in there. And from doing the VSH assay, we kind of noticed that they're in groups, and you kind of see that here a little bit. So Varroa mites prefer drones, and this is mainly because the drones are in the cells longer. The worker is in the cell 21 days, where the drone is there for 24 at least. So there's more time for her to replicate and mate inside of there. But the male mite starts with the sisters and the sperm matures, and the females, and then the male mite dies, and then the new mite will emerge with the foundress. The mated female mite will leave with the emerging bees and probably feed on them for a number of days, and then start the process over. The number produced depends on the length of the pupal stage, and this goes back to the drones, where they've just got a little more time in that cell, so they can here up to 5 mites you can get out of a heavily infested colony, and in workers I've seen the 1 to 2 in there in the cells. So newly emerged mite behavior. While completing maturation, which is basically they're feeding on the nurse bee, and then that exoskeleton is becoming sclerotinized and they're becoming red. Sometimes the mites will feed on adult drones and foragers, and that increases the chance of transmission of viruses by hive drift or via flowers. Eventually, mature female mite enters the cell, and like I said, the cycle just repeats itself, and it's pretty much within in 2 days. They're ready to go. So if there's no brood, they'll just feed on the adults, and this was kind of surprising to me. In the summer, they could live up to 60 days. So if she's going in the cells, even every couple weeks and just reproducing, it's how your mite levels can increase so fast. And in the winter, they can survive up to 6 months in the winter. And coming from Pennsylvania, that's one of the impacting factors is getting your treatments on early enough where you're not treating those winter bees, but also getting rid of those mites, because they will stick around for a long period of time. And the cycle never ends, as I said. People that say that they don't have mites, they're just probably not looking hard enough. So this is just back to integrated pest management approach. And I'm going back to this slide because this is based on thresholds and multiple different treatments or ways to treat the mites. And I'm going to get into, some of these ways that we monitor for the mite shortly here. But Varroa mites, besides forage, is definitely the number one problem. Out of all the times I've been called out to beekeepers, I would say 85 to 90% of the time it's mites. Sometimes it's pesticides. I have had some cases where I think the mite levels got too high. They treated them, but the viruses were there. So sometimes they'll get higher nosema, or they'll get chalk brood from having high viral loads because the immune system is compromised. So it makes them susceptible to more things when you get a lot of virus within the bees in the colony. So mites on adult bees plus mites in the brood equals a lot of mites. So if you're getting a high mite load in an alcohol wash or sugar shake or sticky board drop, there's a good chance there's a lot of mites in the brood still that you didn't get. So these are kind of the phases I would look at. Is just, if you're seeing mites on the adult bees, it's time to do something, and maybe you might be a little too late. It's just, that's why it's important to monitor. We try to do at least once a month. So now I'm going to get into the monitoring methods. You can examine debris on the bottom board. looking at adult bees to see if you see mites. But like I said, they're often feeding on the underside and the sclerites in between there, so you're not going to see them. And then another way is examining the brood, and I'll talk a little bit more about each one of these. But noticing the mites on the bees or picking at drone brood does not count for monitoring. I mean, you know they're there, but you have no idea what level and what kind of treatment you might want to use. So the most common Varroa monitoring methods are alcohol wash, sugar shake, which is not as effective, especially during a nectar flow, because during the nectar flow, when you put the bees in the jar, they're going to be covered with nectar. The mites are going to stick in there, and you could put, a half cup or more of powdered sugar, and you're just not going to get the mites out, especially when they're in between the sclerites. Another method is sticky boards, or mite drop is what they would call that method. And then uncapping brood is not the best way, but it's a way a lot of commercial beekeepers will just check quick to see if they've got a problem or not, and I'll get into detail on that. So for an alcohol wash, basically you're collecting 300 bees, which is about a half cup, from the brood nest area. You try to get it from open brood and sealed brood, so you're getting a varied age, and then place it in a wide neck jar and then close it with a mesh lid, or what we use is a solid lid. And I have some pictures of this whole process coming up. But have the jar filled at least halfway or 2/3 with rubbing alcohol. 70% or higher is what we prefer. You can get away with diluting it down to 40 or 50%, but I don't think it drops the mites quite as fast. And then you shake the jar for a minute, and then you pour the alcohol into the sieve to rinse the bees in a bin to count them and calculate the percentage. But I have some pictures because this is just a brief overview. So what you'll need for an alcohol wash, you need your 70% or higher alcohol, 2 different size sieves, which I have some pictures of these sieves and the sizes on a slide or 2 down if you wanted to look at that, or we can get that information out to you. Paper towels, this is mainly to clean the bins, so it's easier to count the mites. Of course, the jars and 2-way bins and a half cup to scoop the bees. You can pretty much use any container to scoop the bees. Sometimes we cut the smaller alcohol bottles to scoop the bees, and it's easier. You just need to check it with the half scoop and then make a line on the container so you know that you're getting your 300 bees. So this is kind of the alcohol wash process. First, you collect your bees into the jar, and often what we do is, say we're doing 8 in a yard, we'll collect all the samples first and have 8 separate jars. Here there's just 7, but we'll collect all the jars first and then do the mite wash at the end. But you want to shake each one for a minute, and then you dump the bees into the larger sieve, which you can see here. And that enables the mites to get through, but the bees will not go through. And that is hard to see there with the size of the sieve, but I do have some other pictures. And then you have this fine sieve. So if you do get mites in the wash, you put this sieve, the fine sieve, on top, and then you'll rinse the alcohol with the mites through there, and then that will contain the mites, and any other mites that are within that sample will get rinsed out. So this is just kind of repeating what I said, but basically you do this process, and this sample that I processed, luckily it had no mites. But you repeat the process until you rinse it through the fine sieve, or if there's no mites in the first rinse, you just wash that twice, and if there's no mites, it's a 0. Often, if you have high mites, you could rinse 5 to 7 times before you get all the mites out through that fine sieve, which I hope nobody has to do that because it's a bit more counting. But these are the sieve sizes that you would need. The first size is 3.5 millimeters, and that's the one that you do the first rinse through. This bottom left picture where, it keeps the bees, but the mites go right through. And then if you do find mites, you put this sieve on top, which is this ChemLab it's called number 35 or 500 for removing the mites from the rinse. But the thing is, when you rinse the second time, if you put the sieve on, that takes the mites out. So if there's any remaining mites, you can tell that those are new mites that were in the sample, as opposed to just dumping it back through and then you're counting mites that have already been counted. And these can be found on Amazon.com, and I'm sure you could find them anywhere. I would go with whatever's cheapest. And then the bins that you will need, Amazon also has one that's on the right. It's a little more sturdy. The bin on the left can be found at like a Dollar Tree or Walmart, but you could see on the bottom right how it's got a chip out of the corner, and that's just wear and tear. But the one on the right is a lot more sturdy, and it doesn't. You don't seem to have as many issues, and tends to be cleaner. I would say one thing that I've learned about the bins over the years is you don't want ones that have little circles in the corners because the mites can get stuck in there. So it's best to get something that's totally flat in the bottom. It just makes it easier to count. So another method, and I really like the alcohol wash, but when I started as an apiary inspector in 2008, we used to do sugar shakes on 4 colonies out of an apiary. And it's basically the same process, except that you're putting the bees inside of a jar and then you're putting powdered sugar in there enough to coat the bees. And like I said, if there's a nectar flow on, it's a mess. But you want 1/8-inch mesh lid on there, and what you do is I take the scoop that I use to scoop the bees and put it over the screen over the top of the jar and hold those together, and you shake them vigorously for about a minute. And if you're not damaging the bees and the bees aren't kind of messed up when they're coming out, you're not shaking it hard enough and you're not going to get the mites off, and your mite count is not going to be as accurate as an alcohol wash. And as I said, if there's a nectar flow on, the alcohol wash is pretty much the only way to go. And if it's a heavy enough nectar flow, even in the alcohol, you can see an oily looking substance in the alcohol, which is that nectar. And sometimes we'll dump those out if it starts looking pretty oily, because it lowers the percentage of alcohol. But once you're done shaking the mites, then you shake them out on a white container. The opposite side of one of those bins, or inside that bin, is a really good method, because if you shake it inside the bin, if it's windy, they won't blow away. Another way is you could shake them in the water, and it makes them a little easier to count. But that's basically what I got with the sugar shake. So pros and cons on the sugar shake: it's practical, low cost, non-destructive, except for those 300 bees. They do get kind of mangled. It does take time to count the bees if you wanted to count the 300, but it's best to just use the measuring cup and make sure you're getting about the same amount each time. Counting the bees is something I feel that you don't need to do, as long as you're getting that consistent, 300 bees. And then, with that sample of 300 bees, if you get, around 9 or 10, it's time to treat or do something. Alcohol wash, low cost. Water is not practical at removing the mites. And I mentioned the soapy water wash. That's really not practical for removing the mites either. It's better to just use alcohol. And remember to just be consistent between samples. It's best if you're going through 8 colonies, it's best to just have the same person scoop the bees so they're getting the same exact amount, and it's as close to that number of bees that you want for the sample. So now I'm going to get into the sticky boards, and you could do natural mite drop over 24 hours is easiest, but the information that Matt helped me get here is with the sticky board, you use Vaseline and you put a real thin layer on, and this just keeps the fall mites in place, because what happens is the ants will come in and they'll start picking away the mites, or you might have some other ground beetles. Scarabidae is a common one that'll come in and take the mites to eat. And this just prevents other critters from taking your mites away. But basically, I've got this whole list here of 4-day average drops for each month, and what you're looking for as kind of an acceptable level. And you can see that in September, they had treated, and then the average mite drop was lower in October, November, and December. I would say typically commercial beekeepers would treat more in late August, and you wouldn't get that high number in September. But another method, and this is the quick and dirty method on capping brood. You could do 200 cells and, look at them individually, but that's kind of a pain. So what I recommend is at the bottom here, and this is what commercial beekeepers are doing, is you take a sheet of brood and you want it with heads on it, because you'll be able to get the mites, especially if there's any that have reproduced. And you just kind of scratch that open with the sharp flat end of the hive tool, and you scratch all that brood open, and then you bang it out on the lid. If you have dark lids, you would want to use one of those bins, or even one of those the corrugated plastic for the mite drops would work, or any white surface, or even cardboard works. And then you just bang the crap out of that frame onto the cardboard, and then you sift through the brood with your fingers and look for the mites, and you'll be able to tell if there's an issue really quick when you're banging out the brood like that. But doing the 200 capped individual cells is time consuming, and a lot of people are not going to spend the time to do that. And it almost makes sense to look into doing the VSH assay if you want to get that technical with looking for mites. But just scraping open the brood and banging out is the easiest way. You don't need any of the gear, and it's you could tell if there's an issue pretty much immediately. And sometimes it's not what you want to see. If it looks like somebody sprinkled pepper in there, you you got a problem. So pros and cons to sticky boards. Sticky boards, environmentally friendly, relatively cheap, can be reused many, many times. Doesn't really affect any of the brood or anything. It's just the the counting of the mites. That's what takes the longest. It does require having special bottom boards. And you do want to use that Vaseline to try to prevent ants and stuff from taking the mites. And another thing I've learned is make sure that corrugated plastic slides in smoothly into the bottom, because sometimes if the plastic is bowed or if the wood is the window is too tight, it scrapes all the Vaseline off. So when you go to pull the sticky board, it scrapes all the mites off, and then you have to kind of spread that out and try to count them. So it's best to make sure that those sticky boards slide in freely and the middle is not bowed up and touching any of the wood before applying Vaseline or anything, because when you go to pull that out, it's going to scrape all the mites off. A lot of them are going to be stuck on the inside of the wood where you can't get to them. So for uncapping the brood, it's accurate to get an estimate of the mite numbers in the cells. And it can be quite disturbing to the colony because they're going to have to, cannibalize a bunch of that stuff and clean up the cells again. But when a colony is in full force and reproducing, it's not a problem for them. They'll clean it up within a couple days. Not really a threshold on banging them out. It's just, is there a lot or none or a little? And then you can kind of, after doing this several times, you can kind of get an idea of what you want to do as far as treatments. So this is an important graph, which basically just shows, the bees population peak is ahead of when the mite population peaks. So the bees might look really good, and then the mites hit their peak, and then that's when you start getting your PMS and all these other things that pop up. So just remember that the bee population peak is sooner than the mite population peak. So what do we do? You just want to be on top of it. Don't wait until it's too late. And this is why it's important to monitor, because these mites are always on the increase, especially if there's brood. If you have low mites and there's no brood, I wouldn't be too worried. But you might have low mites and there's a ton of sealed brood in there, and the mite wash might show that the mites are low, but it's best to monitor even maybe twice a month if there's a lot of brood in there. Just try to catch after there's a big emergence. Sometimes if you're an experienced beekeeper, you can tell when there's been a big hatch because the bees are just kind of flying in front of the hive and look like they don't know where they're going. And after that is a good time to do an alcohol wash, because that all those mites that were in that brood have just emerged. And it just takes experience to see that when there's been a big emergence and the bees are kind of just fumbling around in front of the hive, just doing their orientation flights. But that's a good time to do a mite wash, just so you're capturing it. Or you could bang out the brood, which is another way. So monitoring is important. And you could see these mites, that's a lot of mites. I wouldn't want to see that in my colony. So general things for consideration: colony genetic, survivor, stock, hygienic, local. That's up for you to decide. I do like the VSH and if you're planning on getting, queens that are supposedly mite resistant, look at reviews, see what other people are saying about them, and remember, when you get those mite resistant queens, it's going to take, at least a month, 6 weeks until those bees are hers. So you're not going to get those traits right away. It's not a immediate effect. And then, apiary size and location. Number of hives can affect your management. If you've got 300 hives in one location, the bees they're not going to do as well. And then you also have to think about your neighbors. How close are they? What are they treating with? It's best to make contact with the neighbor and see when they're treating, and maybe try to coordinate treatments with them, so you're doing it at the same time, and it's your best chance to keep the mite levels low. Think about colony size. Bigger colonies, second year colonies, those are a lot harder to control the mites. So sometimes it's best to split. That'll also prevent swarming. And splitting will give them a brood break, and that helps with mites. And it's one way to try to combat mites without using treatments. And then hive equipment, you can do drone trapping, which it really affects your honey impact. But basically, you have 2 drone frames for each colony, and once she has that all laid up with drones, you could stick it in the freezer for 24 hours, or as long as it's in there for 8 hours, it'll kill everything. And then just put that other frame in there and just kind of rotate them, and that will slightly control mites, but not the best method, and it definitely will lower your honey production. So cultural control and resistant bee stock. This is going into the VSH, or Varroa sensitive hygiene. Usually, hygienic honeybee stocks are good for general virus and clean bottom boards, and they tend to just keep the hive cleaner and give less work to the beekeeper. Nurse bees can detect and remove diseased and sometimes Varroa infested brood. There's several stocks available for purchase. Minnesota Hygienic, the VSH, some of the Russian breeders have really concentrated on VSH. And then there's also the pole line stock from Baton Rouge, but I don't know how commercially available that is, so I did not list that. Or you can try to breed your own and select the low mite numbers, although just beware of your neighbors. If you're trying to select for low mites and you have high mites and you're spreading them to the neighbor, that's you're not really controlling your livestock at that point. But if you're isolated, I think that would be all right. But I'll take any questions, and these are just pictures of hygienic testing, which I'll touch on later. But this is what Matt and I do for potential breeders in Northern California, down into I would say the middle of California even, and sometimes down south if people were interested. But you could see this would be what you want. That's 100% hygienic. And then sometimes you get ones where they just uncap it. And, that's still good. It's just one of the traits, where this is uncapping and removing, and this is just uncapping. but this is the inside after we froze it. But I'll take any questions.
I don't see any questions in the chat. Would anybody like to unmute if they have a question? Hi, Carl. Oh, no, never mind. Thought you unmuted. Do you want to take a break, Rob, and come back and see if there's questions
That's
then?
what I was thinking. I got like another 20 plus slides,
Okay.
and I'll go through the treatments, and maybe have some more questions with that. But with the break, if you guys could just think of any questions you
That's a
have.
dumb question. Feel
No,
free
there's
to
no
laugh.
dumb questions.
Sorry. I terribly operate off of rigid schedules, and I know this is all, say, like dependent on if I'm coming across anything during hive inspections, etc. But say I'm not coming across anything that a simple person, I'm just sorry, hobbyist, very, very hobbyist beekeeper. But say I'm not coming across really much, like see like a K-wing here and there, something else that I could see. But as far as like simple recommendations for say like a timeline, like Central Coast California, if I'm just say inspecting for mites or doing a alcohol wash for mites, like
Yeah.
off the top of my head, I just think of like, okay, cool, like mid-spring, say midsummer, and then mid to late fall kind of
So
thing.
actually, I do have a slide that kind of goes over what the beekeepers up here do, which is maybe it's probably a little similar. How far southern are you?
I'm Central Coast, Ventura County,
Oh,
so
okay,
like
okay.
Ojai, Santa Paula area.
Yeah, so typically, and I'll cover this again later, but typically a lot of the beekeepers we work with again are commercial, but they'll put a mild treatment in January when they first start getting into the hives. You might not get into them that early, but maybe you do. But what they do is they'll treat with a mild treatment like Apivar, which is Amitraz synthetic, or maybe they'll use like a hop guard, something really mild in January, just to keep the mites at bay. But a lot of the guys won't treat again until I would say mid to late May. They'll use a harder treatment like formic acid or even like Apiguard, and that's when they're really trying to knock down the mites. And often for these guys, it's before they go to their honey locations. So the timing of year is usually January, May, and then you really should be looking and monitoring, I would say, early August just to see what's there. And maybe even after May and June, just do like a mite wash just to make sure your treatment work before things can get out of hand. But I would say the most important time to monitor is after you do that treatment in May or late May and see that your treatment worked. And then if it didn't, you might have to switch up to something else. So that's what I would recommend. But the fall's where it gets tricky with the weather, you know. But I would say the most important time to monitor is after you do your treatment in May. When do you usually treat, and what are you treating with?
So right now I'm set up. I have Apiguard and Formic ready.
Okay.
Only tentatively the Apiguard. I wouldn't say ideally not the Formic, but it depends on what I end up coming up with the mite count here probably next weekend. The weather is the biggest thing I'm sort of dependent on. Sorry, I'll answer that in a second. Thank you, Wendy. Just because the other day we ended up hitting like 98 degrees.
Yeah, you don't want that with the formic,
Yeah,
especially with
exactly.
two paths.
But otherwise, yeah. So May, I'm coming up on it. I was planning, I have in my calendar next Saturday, sort of my check, just looking at weather and then my schedule.
Yeah.
So for May and then again, August, and some of that is sort of just weird. I do a lot of like horticulture stuff, mostly fruit trees. So just
Nice.
how I get my schedules and stuff going. But January, I'd not really come up with or
Yeah,
thought about
that's
anything, but we have good weather for it. Usually we'll hit easily. Heck, in January, we probably had a good week of like 70 plus degrees or so. So it's easy for us to jump in the hive.
yeah, and the January time is just mainly for these guys. It's like their only time to treat right before the almonds, because they're gonna really build up in the almonds. So that's a lot of times why these guys are treating in January. But I think just keeping the mites levels starting off the year, and then you won't have as much trouble in in May. They won't be as high, and a formic treatment at that time of the year. The only issue you run into is if you lose queens, then what, there's no way to get queens at that time with the formic. So that's why guys use like a more mild treatment at that time, like the Apivar or Hopgar even. And hopefully we'll get Varoxan legalized here. And I'll talk about the treatments quite a bit more here later in the presentation.
I don't want to jump ahead too far or steal the break. It looked like there was a little asterisk next to the OA. Are you going to, by chance, I have random questions about that, the thought of it going forward, if anyone knows of more
Yeah, so
speculated timelines on OA being allowed in California. But I
yeah,
can argue
so
about that later.
so Alina is working on this white paper to try to get this ball rolling. And Matt, chime in here if you know any updated stuff. But right now, I just don't know. I'm hopeful for it. I mean, I think that would be great treatment to use in January. I mean,
Yeah, I would agree with Rob. I'm recommending. I'm in Colorado right now, and it's a great treatment. This Varoxan or oxalic uh, in the summer because you're allowed to use it while honey supers are on. So that's key for us. That we're making honey, but we need to keep the mites low. It's. I would add that it's a good one if your mites are already pretty low. Oxalic will keep them low, and this Varoxan product. Alina has written a letter to the state. Last I heard, it was tied up in approval processes. I feel like this is just it's got to cut through this red tape. Basically, California is one of the most rigorous and regulated states. Oxalic has been approved since what 2014 or something for the rest of the states.
yeah.
So yeah, she has written letters. She's worked with the CSBA. It's just really hard to get government in California to approve things. They're well, yeah. It's in process. I can't give you an ETA. I would hope in another year we should see it legalized. This is true also for the new product called Amiflex, which Rob will probably talk about.
Mhm.
It's a heavy hitter, miticide, but same thing. It's not yet approved in California, where it's approved in most other states. One thing that would help is if you're in an organization down there, your clubs, if a club would write a letter, and you can we can ask Alina who to write your letters to, but it is kind of frustrating that we're not able to buy this in California.
Yeah.
Thank you,
I
Matt.
mean, if you are interested in reading more about oxalic acid, Randy Oliver's website has a bunch of stuff. not recommending it. It's just it's information on it. And you could see some of the stuff that he's done with it. But until it gets legalized here, hands are kind of tied, and you're stuck with the APAVAR, and then there's APAVAR 2.0, which I tried to research and figure out how it's different than the original one, but I haven't figured that out, and I'll talk about that later. But I am gonna, unless you've got another question, I'm gonna use the restroom quick.
No, that's great. I appreciate you. Thanks again, also, Matt.
Yeah, and if you have more questions at the end, I'm gonna take questions again. So
Awesome,
feel free
thanks.
to ask anything. No dumb questions. We're all just learning. I learn something new every day with bees. So I think it's an endless supply of information.
For sure.
Yeah, let's come back at 11, shall we?
Yeah.
Sounds good.
Nice. Welcome back.
Yeah, I was gonna say if anybody had any questions, I could go back to previous slides or anything like that, if there were other questions.
I had an observation. I I was out visiting some friends over in Virginia City, or actually Nevada City. Sorry, here in California, and kind of stumbled upon Randy Oliver's garden, bee garden, and he was out working bees and making splits. And I noticed I was chatting it up with him a little bit, and noticed his record keeping on his hives, and his Varroa account
Yes.
was there, and it was many zeros, and then there would be a one or a two. And I know that he uses certain methods for Varroa control, but I think he's also trying different things with genetics within his apiary in his business. You have any comments on that? And because I mean, I was just seeing 0002.
No, so he's really been working on, the low mites, and I'm not sure where he got the stock or if he just selected himself out of that. I know he's been working on a lot of this stuff for a number of years, and he does use some methods that I can't really talk about, but uh, I think he's mainly concentrating on, bees that take care of themselves and don't really need much mite intervention, which I think is great. Um, I've looked at stock from Hawaii that was pole line several years ago, and they weren't quite up to par as compared to a commercial beekeeper stock that was run, treated, fed the same way. But there is a pole line coming out of Baton Rouge, and I don't know, Matt. Maybe you know more about the commercial or even public selling of that stuff. I don't think it's online yet.
Yeah,
But
you can't get them from Baton Rouge directly. You know, that's the USDA.
yeah,
You can't really, not allowed to sell queens per se, but they have worked with a number of breeders, smaller breeder shops where they're selling, instrumentally inseminated queens to them, or distributing them. They can't sell anything. And you can find pole line genetics, from breeders in, American Bee Journal or online. And yeah, Rob and I have studied the Hilo queens, and we're doing a study now with the pole line queens, and it's legit. The whole VSH thing where they're doing the very tedious assay of teasing out, plucking, as they would say it, out uh, 200 pupa, checking each cell individually for a mite, and then if there's a mite, checking if that's a reproductive mite or a non-reproductive mite. It's legit. These Hilo and pole line queens, they will keep your mites lower. They're not going to be totally treatment free. They're definitely could be a big part of your IPM strategy. But Rob was alluding to before is they're not up to par in terms of honey production and spring buildup when compared to some of these, Italian lines from Northern California. So, it hasn't really caught on for commercial guys so much. These guys need to make money by, spring buildup, selling bees or honey, or just being big enough to make pollination contracts. So they would rather, treat their bees with a miticide and have them build up faster and then use some of the other Hilo or pole line queens. But I think they're great. Especially for hobbyists or sideliners who don't need to pay the bills with their bees. It certainly is a great way to help with your bees just surviving. I mean, I'd rather have my bees alive and small, in the spring than, a big one that has a huge pile of dead bees on the bottom board, because, mites killed it over the winter. The winter bees weren't healthy or whatever. And uh, but yeah, the VSH thing is legit, and I see a lot of people advertising for it, but buyer beware. Just because you buy a VSH queen a few years ago doesn't mean that your queens you're selling are VSH. You need to be on top of it and buy an II queen every year, or do your own work and do your own VSH assay and selection process, which is what Bee Health Hub started doing this year for the commercial guys. It's just a very tedious process, and we were able to find a little bit of VSH traits, but it's going to take a few years for us to develop this with the bigger commercial breeders. And as far as Randy's, his methods and he's not doing the VSH assay. He's kind of going more old school, and he's just doing mite washes, and anything above a 2 he'll take out of his breeding program and he'll throw a treatment on. But he's mite washing every colony. He's got, I don't know, between 1 and 2,000 colonies in their operation. That's a lot of mite washes. The average beekeeper can't do what Randy does. Of course, we all know Randy's not the average beekeeper. So he's mite washing every single hive, so, more than once a year. And that's how he's selecting his. And he doesn't care whether they're, the VSH hygiene or ankle biters or whatever method they are for keeping the mites low. He doesn't care. He's just selecting by the actual results, which is the mite. And he does a mite wash with alcohol. So yeah, I haven't actually tried any of his bees lately. I know some people that are getting a few. Would be interesting. I know he said that it's taking longer and it's very difficult to breed for this, and to keep a you know the attributes all the commercial guys want, right? Early buildup, lots of honey, just big, strong colonies, which is, to me, my intuition, it's like, yeah, well, the big, strong colonies are the ones that have the most mites, right? Because they have the most
Yeah.
brood. So talking to Frank Rinkovich, I just asked him, he's leading up the VSH project for us, and he's from Baton Rouge. And, I said, what is there a trade-off? If we want VSH bees, do we have to accept lower honey and slower buildup? And he said, yes, there is a trade-off. We just have to find that sweet spot. I know there's been some VSH bees. I ran Hilo for a couple years where they had zero mites, but they never got bigger than one box of bees. Never made me any honey, and they were almost too small to go through winter. So you can even go too far extreme VSH. So yeah, that sweet spot is difficult to hit. What are people willing to give up? I'll give up a little bit of honey, but
Yeah, and Garrett there, he's been doing inseminations, and they really started concentrating on this in 2012. So it's relatively new, but not that new, because they've been doing it for well over 30 years. I know Harpo's done a lot of work on it, and it's just commercial guys are now starting to be interested in it now that we can do the assay. And we did a bunch this spring, but the problem is with the commercial guys is they're always trying to keep the mites low, and especially these queen producers, they have some of the lowest losses out of anybody because they have to replenish all the dead outs every year and sell all these queens, so they can't let the mites get too high to allow us to do some of this stuff. So it's trying to convince them to maybe select a yard or two where we could do this, where they at least don't treat them for over 3 or 4 months would be better, but they recommend 3. But these guys are really good at controlling the mites. So this spring when we did the assay, I mean, the mites were so low that it was hard to even find mites in the cells, let alone reproductive or non-reproductive. We found a handful, but I think doing this later in the fall before they treat is going to be our golden ticket to maybe finding some of the stock within these commercial queen producers that they already have there. It's just getting them not to treat, so we can actually find mites to figure out if they're uncapping and disrupting the reproduction. But any other questions on previous slides? I did want to say with DWV, something that I find interesting is almost every time that you find a bee that has the twisted wings or crippled wings, if you pick it up, there's almost always one mite on it and sometimes two. And if you're lucky, you'll get three. Not really lucky, it's unlucky. But yeah, sometimes, just picking up those bees and looking and knowing what a mite looks like on a bee and where it hides. And it hides in the sclerites and on the underside of the bee. And the mites also, they don't like sunlight. So as soon as they see light, they crawl to the underside of the bee. So if you're telling me you don't see mites, it's like, well, those mites are not trying to be seen. They're hiding on the bee. And a lot of times when they're feeding, they're in between the sclerites. So the only way you can tell it's there is there's a lump on the underside of the bee. So visually looking for these things is unless you've got a really keen eye, it's really tough. And even those parasitic mite syndrome pictures I showed you, I might have saw one or two mites and took the picture, and then you start zooming in and it's like, holy crap, there's 7 on one slide, hiding inside there. And yeah, it's just amazing what the eye doesn't capture, but what a camera can. So snapping a couple pictures here and there and then zooming in later. I mean, the phones nowadays are incredible. I recommend taking a photo in the shade with the flash. That's how you're going to get the best picture. Getting it inside the cells is very hard. I use a digital SLR on manual focus, and that's how I get the clarity down in the cells and can get that depth. But that's with it's a $600-700 camera, so a little different than a phone. But the phones have come a long way, and Harbo, I think, Matt, correct me if I'm wrong, but he's been using the cell phone to do the VSH, or one of those guys is.
Yeah, it's Harbo. He likes to use his phone. Yeah.
But if we're ready, Keon, just let me know.
Yeah, I think we're ready. A lot of great information here. And back and forth. I think we're ready, and we're already recording, so feel free to start when you're ready.
Oh, sure. So I just wanted to do a shout out for me, Matt, and Keon and Wendy's helped out with we started this 2 years ago. We made a website, and we've got a bunch of different services. If you guys are interested in taking samples and sending them in, we could do Varroa and Nosema on those, and all the pricing is on this website here. But Matt and I worked for Bee Informed Partnership. I think, Matt, what were you there, 5-6 years? And then I was there 12 years. Kind of started off, Katie Lee started, and then Mike Andre and myself started in June 2011. But this is some of the information for the hygienic testing. And this was from 2010 to 2020, but this is some of the results of all each of these different colors is a different beekeeper. And what this is showing you is the hygienic scores over time. And you'll notice a lot of them they go up. And doing the selection for hygienic, it increased over time. And this was something I had our statistician, Natalie Steinhauer, look at. And you'll notice a lot of these went up, but not only did they go up, but they were more consistent overall when we sampled them, which means that when we sampled the group, that overall the percentage was going up, not just a couple colonies. And there's another graph here which shows you the percentage of colonies tested which scored 95% or higher. And you'll notice there is some variation here, but some scored really high. But for me, doing this work for so long, it's like, did it make a difference? And then when she looked at it, everybody thought I was crazy, and didn't think that, it would have been increasing the way it was. But, after about 3 years, if you select hard for it and have isolated mating yards, your percentages will go up. And maybe at the end, Matt can talk about, he started this with a couple beekeepers, and I know that he's seen a great increase with the beekeepers that he started the testing with. In the beginning, they don't test very good, which we don't like because it's a lot of counting. Where if they test well, it's you don't have to count as much. Like in that question slide, you want those zeros because easy to count. You're done in 5 seconds. But if you got to count 100 cells, it's a bit more work, and it's really draining on the mind after doing it, especially if you're doing 100, 104. I think, what did you do, Matt, this year? It was like 120 or something like that.
Yeah.
You did. But here's the big one. This is pretty much the guts of this whole thing, is the treatments for mites. In California, this goes back to the question the gentleman asked before the last break. In California, most beekeepers or commercial beekeepers and queen producers will use a mild treatment in the spring, like Apivar strips. They really like those because it can rain, it can do whatever with the weather. It doesn't seem to affect those strips, and it's not hard on the colony. The next time they'll treat is usually in late May, and they're also usually requeening right before this, or they'll use formic acid and they'll hit the bees real hard with the 2 strips, and then they'll come back and requeen. So they if they knock a queen out, they don't have to worry about requeening because they were already planning on it. And it is for commercial beekeepers, the practice is pretty much to replace the queen each year because she's more vigorous, less likely to swarm, and it's easier to maintain. But they use treatments like Formic Pro at this time. Apigard is another one that they'll use. Long as it's not too hot, Apigard can, if it's 8 frames or less and you put a full dose of 50 grams on a colony, it's most likely going to abscond. Even with the Formic, if you put that on, they might beard out if they're big, but you don't want to use 2 pads on an 8 frame, or but just follow the label. It kind of tells you what to do by how many frames of bees are there. And then their next treatment is usually late August to September, unless monitoring mites, they're higher before that time. Then they're going to treat. And I'm just going to point out these pictures. This is a Cordovan queen. These are sold by Frank Pendle and the Conans. It's a kind of, they call it a redhead. It's got almost like a reddish color thorax. They're a cool looking queen. Really big populations. Definitely eat more than normal Italian. But if you want to make packages and things like that, that's where this bee is an advantage. And then you'll see here, I wanted to point out this picture. This is something, it looks like a disease, but this is a colony that is not hygienic, that had parasitic mite syndrome. And this is about 3 to 4 weeks in. where you get these secondary bacteria moving in and breaking down the larva, and it makes them look kind of funky like it's disease, but it's actually parasitic mite syndrome. It's just that it's old and the bees aren't removing it, and it's probably because they're sick and the population is dwindling. And you'll see in the bottom left here, this is a picture of that frame where you could see how spotty it is. And that's one of the ways to know if your colony is healthy or not. You want to see a sheet of brood. If you see something spotty like this, or even if there's a little bit of spottiness, it's better to look at more frames than less and try to figure out what is causing this spotty brood pattern, because often it's a disease, mites, or you could see this colony, there's pollen around the outside, so they've got food, but there's not much honey. So maybe there's a nectar shortage, and then sporadic nectar flow is something that causes European foul brood. So again, I'm not going to talk about diseases, but it's important to notice that there's no honey in that frame, and that could be the culprit of why this colony is breaking down. But this was actually from mites, and they were pretty much not doing so well. And you never want to see a brood pattern like that in your colonies. If you do, it's an issue, and if you don't get it, you could take pictures and send them to the Bee Health Hub, and we could try to identify it. But again, use the flash and take the picture in the shade so we can get a good look at the larva. If it's blurry, we can't tell what's going on. They need to be clear pictures like this to be able to identify what might be happening. And knowing the hive history and what you did is important too for helping diagnose what happened to the hive. So this is a list of current legal treatments. I'm not sure that Sucricide or Sucrashield. I know Sucrashield, I have not seen that. I tried Sucricide, but this was like in 2008 or 2009, and I didn't like it. But you can see here what treatments you can use with honey supers. So Hop Guard, Mite Away Quick Strips, and Mite Away Pro or Formic Pro. These are the only things you can use currently. If oxalic acid is added to the list, that would be another one that you could use with honey supers on. But you could see here on the right, what is allowed in California, which we're limited. And I'm going to talk about a couple more that are limited and kind of go into detail on some of this stuff. Apistan is one that a lot of beekeepers used when it first came out, and it worked great, but there was a great resistance in it. And I've heard of beekeepers using it nowadays, and it had effect and it was working. But I know in California, some beekeepers tried it and it still didn't work. So that one I would just stay away from. But now let's get into some of these into a little more detail. So HopGuard 3, this is the third formulation. I looked at this stuff way back in 2009 and 2010 in Florida, and it didn't seem to do that great of a job. And that was compared to Formic. And the one thing I did learn is formic acid draws in hive beetles, and there's so many hive beetles in Florida. We're lucky we don't really have the hive beetle problem here, but if you are in a hive beetle area, just beware that formic acid, it stresses the colony, and that's what the beetles concentrate on, is the pheromone that the bees put out when they're stressed. So when you use formic acid, the beetles can sense that the bees are stressed and will actually go to that hive with the formic, which I'll talk about in a minute. So this is organic treatment. It's a extract of thyme, which is a plant. And this is another one to use in May before you're requeening in case it knocks any queens out. And again, you may want to put a little bit less on if it's 8 frames or less, and the bees should be fine and won't swarm or abscond, I should say. And it's kind of a less impact treatment, but if you use this and it's hot, the bees tend to cannibalize the brood. So that is something to keep in mind. Basically, you use this treatment 2 times, about 7 to 10 days apart, and they come in 50-gram trays for a hobbyist. If you're a sideliner, you can get it in a tub, and they give you a syringe, and you can apply the correct amount on a card that's similar to this metal. It's basically a piece of paper with one side that has like an aluminum foil on it to prevent the bees from chewing it up. So Formic Pro is basically one of the treatments that a lot of beekeepers are using right now. It's the only treatment that kills mites under the capping. So this is a good one to use before requeening. It will knock out old queens and failing queens. So this is an issue with using it as a fall treatment, is if you can't get queens and you knock something out, then you're going to end up having to combine. So keep that in mind. It definitely has more of an effect on older queens. That's why a lot of these guys that use Formic, they're replacing the queen each year to prevent queen loss. But this is a good one to use. And gentleman earlier thought it might be good in April, which I know this year, Matt and I, we treated our bees coming out of almonds with Formic, and it did pretty well. The temperature was good. Early to late May is another good time to use this. You can use it in August and September. Just be aware of the queens, and maybe have some nukes or something to fill in any colonies that the queen gets knocked out. But yeah, there's just some of this information, these supersedure cells. This is to remind me that if you're seeing swarm cells in late September, and a time when they're not wanting to swarm, it's usually a sign you got a mite problem. So yeah, I don't know. Sorry about that. That slide got mixed up. But Apivar, this is what I talked about for using in January in California. This is good to use when mite levels are low, 3% or 10 mites out of 300 bees. It's not a strong treatment. It will not work on high mite levels. I mean, it'll do a little bit, but it's not going to knock them down the way you need, like a Formic or for Apiguard, or a treatment like that. And they're also good as a follow-up treatment in the fall. If you were to use a Formic or an Apiguard in the fall, and then to put these strips in after, that just that enables you to have that long, up to 52 days of extra protection. Because what happens in the fall is a lot of the commercial beekeepers that are coming back from out of state from honey production, if you're near any of them, they may have mites because when they're chasing the honey, they might not have gotten back to treat as soon as they wanted, or it rained and they weren't able to treat. And whenever you have a big influx of bees coming towards your bees, it's best to have some sort of little barrier to help prevent those mites from wanting to come in your hive. And especially if they bring back something weak, and your bees are robbing something out with mites and picking them up, you've got that treatment in there. So you got that peace of mind that you've got the treatment in there. You're doing everything in your due diligence to keep your bees as clean as possible going into the winter. So this is a new treatment. and Matt and I have looked at this, and this was in the past year or so, and it looks really promising. But the problem is it's not legal in California. It's called Amiflex, and it's ready to use, maximum convenience. You basically apply it on a wooden tongue depressor, and you come back and you do that about 2 weeks or so apart, and it works really well. It's just it's not legal in California. It is amitraz-based treatment as Apivar, made by the same company, but this is 2% amitraz, where the Apivar is 3.3% amitraz. So the strip is actually higher, but the delivery method on this is different, and it works more effective on higher mite levels. So hopefully this one's legalized soon for us to use. So when will this be registered? So this was pretty much the same slide if you guys saw this presentation last year. They're still working on getting this legalized, and Alina is working on this white paper that will hopefully get this product registered in California for us to use. And then there's one more that I think would be really great if they release this. This would be something you would use in January or after you treat in the fall, to just put something in there to have a peace of mind, because this stuff takes a longer time to work, 50 plus days, and it knocks the mites down, and it's really non-invasive. It's oxalic acid from the plant Oxalis, the genus. So you put basically these strips in, and they're cardboard. And then what you want to do is, after 2 weeks that the treatment is in, you want to move these strips around, and it'll actually make the treatment work more effective. And I forgot to mention that with Apivar, is you could do the same thing. After 2 weeks, you want to go in, scrape any propolis or wax off those things, and relocate the strips, because the bees will kind of move away from it. They don't really like it, but the bees need to be next to it and have to have contact to kill the mites. So moving those strips at 2 weeks is important part of using this treatment, and I believe it definitely makes it more effective. And this is kind of what they look like. They're pretty long strips. You fold them over, and then it goes down in between, right next to the brood. And sometimes they will chew down the brood underneath, but they do the same thing with Apivar. You'll get little lines in your wax, but in a couple of years they'll just patch it up. But thank you so much, guys. I finished a little early here, but I'm hoping that some people have some more questions. Feel free to have me go back to any slides or anything you guys are interested in here.
I got a quick